Greetings, everyone. Once again, I want to say welcome to my YouTube channel. In today's class, we are going to be talking about a protozoa parasite that is called Giardia lamblia. I've talked about protozoa, and I think we have studied and uploaded the content for all the protozoa parasites. I know I've talked about Entamoeba isolatica, talked about Trichomonas vaginalis. Sorry, I've talked about Trypanosoma. I've talked about Plasmodium falciparum or SP, then Balantidium coli. So the only parasites that I have not talked about or we have not studied is the Giardia lamblia, and that is what we want to do in today's class. When it comes to protozoa, you discover that protozoa are made up of different categories. We have the sarcodines, we have the amoeba, we have the ciliata, where you have the Balantidium coli. Then, of course, we have uh, the flagellata, where you have the Giardia lamblia and the Leishmania and Trypanosoma. Both Leishmania and Trypanosoma share the same suborder called Trypanosomatina, and they are called the hemoflagellates. As a matter of fact, we have three different types of flagellates in protozoa. The hemoflagellates, and like I said, the examples are Leishmania and Trypanosoma. Then we have the tissue-dwelling flagellates, such as the Trichomonas vaginalis. Then, of course, we have the intestinal flagellate, which is called Giardia lamblia. Now, they are called or they are given a name based on where they dwell within the body of the host. So, for the case of a Giardia lamblia, the basic characteristic is that they are bilaterally symmetrical. That means that this parasite can be divided into two equal parts. That is what we mean by being bilaterally symmetrical. It is also important you know that this parasite also looks like an avocado pear split into two different halves if you are looking at it. And they're equipped with about four different pairs of flagella. So this is the first pair of flagella, the second pair of flagella, the third pair of flagella, and the last pair of flagella. They're also equipped with suckers. By this side where you see the nucleus, you see that they're equipped with suckers, and the function of these suckers is to actually help the parasite to cling to the wall of the small intestine where it dwells. And of course, they have a pair of nuclei. That means they have two nucleus. So this is how the trovozoid of this parasite looks like. But the cyst does not look like this. It's important you know that this parasite exists in two different forms. It exists in two different forms. It exists as a trovozoid. It can exist as a trovozoid, can also exist as a cyst. So what I drew is the structure of the, the trovozoid. Now for the cyst, you discover that the cyst is said to be quadrinucleate. The cyst is said to be quadrinucleate. This is how the cyst looks like. This is how the cyst looks like. This is just a sketch. So the cyst, the basic characteristics of the cyst is the fact that it is quadrinucleate. What does it mean to be quadrinucleate? It means that they have four different nuclei. And these four different nuclei are gotten when the parasite is moving towards the colon within the ileum of the small intestine. Where incisation occurs. So before incisation occurs, then the two different nuclei of the trovozoids will undergo division to form four different nuclei. And once the four different nuclei is formed, then that is when the process of incision is completed before the parasite can now live through the feces to the external environment. Because if a cyst is not formed and the parasite lives, then of course it can be damaged or it can be killed through adverse environmental conditions within the environment. And that is why it forms cysts, and that's why the formation of cysts is triggered so that to protect the parasite when it eventually leaves the body of the host. So I've mentioned the fact that this parasite exists in two different forms. It can exist as a trovozoite within the body of the host, then it exists as a cyst outside the body of the host. And I've given you the reason why it exists as a cyst outside the body of the host to avoid the parasite from being destroyed by adverse environmental conditions. The trovozoite is the invasive stage where it begins to cause different harms to the body of the host. Then there's what we call encystation and excitation. I think I've explained this when we were talking about encystation, when we were talking about entamoeba isolytica. But let me just say it again. Encystation and excitation. So when we say encystation, that means the parasite encysts. There's a formation of cyst, a kind of shell that covers the parasite, the trovozoite. So if we have this as a trovozoite, so when encystation occurs, that means a shell or a cover covers it. Then excitation is the breaking of the cyst. So when the cyst is ingested, the cyst is broken. So the breaking of the cyst is what we call excitation. Having known this now, then of course classification of these parasites. The kingdom Animalia, then phylum Protozoa, then class subphylum Sarcomastigophora is a long name. Then class is Zoomastigophora. Zoomastigophora. So let me rewrite this Sarcomastigophora so that you'll be seeing Sarcomastigophora. This is the classification of Giardia lamblia. Then the next one is order Diplomonadida. The family Diplomonadidae, then genus Giardia, then species lamblia. So this is a classification of this parasite. Kingdom Animalia, phylum Protozoa, subphylum Sarcomastigophora, class Zoomastigophora, order Diplomonadida, then family Diplomonadidae, just add E, then genus Giardia, species lamblia. So having known this now, the next thing to do is to talk about the life cycle of this parasite. The life cycle of Giardia lamblia begins by the ingestion of the infected cyst. When the infected cyst is ingested, of course, it could be through vegetable, a contaminated vegetable, or maybe through the contamination of the food of the host. If the feces or someone who is suffering from giardiasis is used as manure in the farm where vegetable is planted, and of course, if you contaminate the vegetable, and the individual comes home without properly washing the vegetable, then the person may as well consume or take in the cyst of this parasite that has contaminated the vegetable. Or let's assume that somebody is suffering from giardiasis. The name of the disease is called giardiasis. Giardiasis. So let's assume that somebody is suffering from giardiasis, and a mechanical vector such as Musca domestica, that is the housefly, perches on the feces and now perches on the food. And now comes to perch on the food of such an individual. Of course, that's another way that one can take in the cyst of this parasite. So, whichever be the way or whichever route, let's assume that somebody has consumed a food substance containing the cyst of this parasite. So what happens is that once it gets into the body, within 30 minutes of consumption, the cyst begins excision. And when I say excision, like I explained, that means the cyst breaks. When the cyst breaks, the trovozoite is released. And don't forget that this trovozoite is equipped with two different nuclei. So by the time the trovozoite is a cyst, the trovozoite begin to cause harm to the intestine. Of course, it uses the two suckers to clench itself on the wall of the small intestine, and that is where, of course, where it is a cessation, of course, a cessation, of course, within the small intestine. So it begins to cause havoc or harm within this intestine. When it is done, causing this havoc, or probably when some environment, some internal factors of the host, trigger the process of excision, then the parasite begins to walk towards the colon. As it is walking towards the colon, when it's still within the ileum, it begins the process of excision, and that process of excision means that there will be a division of the two nuclei. One nucleus divides into two, the other nucleus divides into two to form a total of four nuclei. And once these four nuclei are formed, then that the process of excision is completed. That means a cyst or a shell-like structure or a cover covers the trovozoites. And once a cyst are formed, they don't become a trovozoites within the same host. So what next is for the cyst to pass out through the feces to the external environment, and the circle continues. So what I just said now, maybe I can use a diagram to explain the structure. So this is environment. So within the environment, we have the cyst, the infective cyst, which is the infective stage. The infective cyst. Then this is infective cyst. We now have ingestion. This is not ingestion. Then excitation in the small intestine. Excitation in the small intestine. Then what again? Trovozoites. Trovozoites are released. Then when they are done causing harm, then excitation occurs. Then of course, cysts are released into the environment. Cysts are released into the environment, and the cycle continues. So, like I explained, this diagram can also help to give you, it's a very simple life cycle. The life cycle is very, very similar to that of entamoeba solutica. Just that the that of entamoeba solutica involves the large intestine, unlike this one that involves the small intestine. So having noted this now, pathology, what does this parasite cause? Of course, it causes abdominal pain. It causes abdominal pain because those suckers, of course, are used to clench on the wall of the intestine, and of course, it can cause slight injury or hemorrhage within that region where the suckers have been used to clench on the wall of the intestine. Then, of course, the person may begin to experience a diarrhea, or even nausea. They begin to vomit and weight loss. The person also begins to experience a weight loss and what we call malabsorption, because this parasite uses its body to block the surface area from whence absorption occurs within the small intestine. That region within that where absorption occurs within the small intestine, the parasite blocks it. So it reduces the rate of absorption of nutrients, and that is also one of the reasons why the person may begin to experience weight loss, because nutrients are not absorbed as it should, because the parasite has blocked the region where absorption takes place, the absorptive region. Then that is some of the pathological effects of the parasite. Then, of course, when it comes to diagnosis is through the demonstration of the presence of cysts in fecal sample. So you get the cysts of the individual, and a kind of investigate the cysts to see if you can find the eggs of the parasite in the cysts. Students who are studying med lab or med lab scientists, they have been taught of how to, how to identify and recognize the disease of these parasites. So if you are a med lab student, of course, by the time you graduate, you should be taught or should know how to specifically identify the eggs of some of these parasites. There are a few characteristics or a few things you can use to identify the eggs of this parasite, but I don't want to go into that. Then, when it comes to treatment, metronidazole can also assist. The same metronidazole that we use in the treatment of amoebiasis can also serve in this case. Then, what again? Prevention and control, personal hygiene, personal hygiene, personal hygiene, personal hygiene. So, personal hygiene is critical, and a human feces should not be used as manure. Then, people should also wash their hand, when they are done going to the toilets. Then, of course, when you're back from school or from work, wash your hand before you take your food, because you don't know if the bus conductor who gave you change is actually suffering from giardiasis, because circulating back notes can also be one of the epidemiological factors that help to spread the disease. So in terms of epidemiological factors, circulating bank notes, like I just mentioned now, yes, food vendors can also spread the disease. So if someone who serves food for you in a restaurant is suffering from giardiasis, the person went to toilet without properly washing their hand. If the person uses the hand, some people when they want to carry food, this finger is usually goes inside the plate. So that's if that finger is contaminated, then with the feces, with the eggs or with the eggs of the parasite, yes, that is found in the feces. Of course, the person defecated now, the eggs or the cysts are always found within the feces. So that hand can also bring about contamination. And like I said, mechanical vectors such as flies can also help to spread this disease. I think this is all I have to say when it comes to Giardia lamblia. We'll talk about other parasites in subsequent classes. Once more, thank you for being part of this class. I am Dr. Anhujon Ujena. That reminds me, if you know you have not been subscribing, you have been watching these videos and you have not subscribed, what will you gain? Just hit the subscribe button. It's as simple as that. And if you know you like what we're doing here, of course, you can also help to share the video to other medical students, not just around the Enugu states, other states of Nigeria, where you have friends that are studying parasitology. Of course, you know that this content will help them so much. All right, thank you.